So what we presented was a series of results that have to do with how we can complement the existing paradigm of oncogene addiction and immunotherapy by using data from RNA sequencing from cancer patients that can be useful in predicting the specific drugs that these patients will be responsive to. So we started from a very simple observation, which is that really what determines the state of a cancer cell is not the mutations, but actually the activity of the proteins. And the presence of mutation in the corresponding genes is one possible way in which the activity of these proteins may be dysregulated, but it's not the only way. There's all sorts of things. For instance, you may be exposed to estrogen through normal sort of hyperproduction of the hormones, or maybe through the pill for all your life. So you don't have an actual mutation, but you get aberrant signals that then contribute to the activation of the corresponding protein downstreams. So based on that observation, we decided that wouldn't it be nice if we had a way to actually directly assess protein activity instead of having to infer it potentially from the presence of mutation. And we developed an algorithm that has just been published in Nature Genetics, which has also been licensed exclusively to the Darwin Health Company, which allows us to essentially very accurately estimate the activity of a protein, and in particular the differential activity, whether there's been an increase or a decrease in protein activity, from the expression of the protein targets. And we have generated what you would call a multiplex gene reporter assay for each different tumor type, about more than 30 different tumor types. And so that we can now very accurately estimate the aberrant activity of about 6,000 proteins in more than 30 tumor types. Well, you can bring it to your clinic and put it to use right now. We are working very hard to have within the next 60 to 90 days a CLIA, actually New York CLIA certified version of the test. We have two tests right now that are generated from this approach. The first one is called Oncotarget. And what Oncotarget does is essentially almost like a straightforward complement to what you would normally get by a mutational analysis. That is, in a mutational analysis, you would get a list of the genes that are mutated, and there are therefore potential candidates for therapy using the corresponding inhibitors. In this case, you get a list of proteins that are aberrantly activated among the proteins that we already know how to inhibit with an existing drug. So for instance, it would tell you that HER2 is activated in this patient, whether or not there's a corresponding mutation in that particular gene. Okay? And the value there is that we see a number of patients that harbor absolutely no mutation and yet have totally aberrant activity of the corresponding protein. So for instance, about 15% of lung cancers have activity of EGFR that are comparable to the patients that have the worst activating mutation in exon 20 and exon 19, et cetera, and yet have completely wild type EGFR, right? And so we would expect that these patients would actually be very good candidates for therapy using Tarceva or another, afatinib, another EGFR inhibitor. So that's where this is helpful, because while we normally find really actionable mutation in only about 25% of all cancer patients that present in the clinic using typical mutational landscape assays, using Viper, we typically find somewhere between 5 and 20 actionable proteins in 100% of cancer patients. Yes, actually, the real issue is not that much one of restricting things, but actually of having a slightly broader repertoire of choices. Because, for instance, as I said, 75% of the patients that we see in the clinic literally have no actionable mutation. They may have a KRAS mutation, which is considered partially actionable because you could use a MEK inhibitor, but MEK inhibitors don't really work very well in KRAS mutant patients. You may have a p53 mutation, and you have bevacizumab. And again, that's not a drug that cures from the cancer. It may delay the ultimate untimely death. So the idea here is that having a broader repertoire of potentially actionable proteins that can be also associated with an inhibitor gives the oncologist a little bit more choice. And then, of course, these potential actionable targets need to be then vetted through a rigorous clinical study process. And so we are already designing collaboration with some of our major partners, MD Anderson or the Rossi, et cetera. We are already designing potential clinical trials where we would actually do the equivalent of what is called a basket study, that is, identify what are these targets that come recurrently as being very highly activated in patients with a particular type of cancer, and then basically designing a study that would test the corresponding inhibitors across the repertoire of targets. So that's the first technology, Oncotarget. The more exciting technology, I think, is the one called Oncotreat. And what Oncotreat does, it starts from a very simple question. It says, how is it possible that when you look at tumors that have wildly different genetics, they actually have almost identical transcriptomic states? Okay, and so for instance, if you take triple-negative breast cancers, mutations are all over the place. Almost no two patients have the same set of mutations. And yet, when you look at their transcriptional profile, their spread is not very different from the spread of the normal tissue. Okay, so that's surprising. It means that the cancer cell, is actually very tightly regulated. And so it is legitimate to ask, okay, so if this is this incredibly large landscape of tumor mutations that we see in triple negative, and yet the state of the protein is so tightly regulated, then there's got to be a bottleneck. There's going to be some proteins that are responsible for maintaining that state. We call those proteins master regulators, and we call the little modules that they form, literally anywhere from 2 to about 20 proteins, we call them tumor checkpoints. And we've shown that these tumor checkpoints really exist across every one of the tumors that we see in TCGA. And that collapsing this tumor checkpoint, either genetically or pharmacologically, induces dramatic tumor regression in vivo. And so what we're now doing systematically, we can take an individual patient sample or even a single cell from a tumor, and we can identify what are these proteins that maintain that tumor state, the master regulators. And a very big surprise is that most often these are not the oncogene. The oncogene are actually further down in the list of the most activated protein. The ones that are the most activated proteins are the ones that maintain tumor state. And then what we do is we basically test the entire repertoire of FDA-approved and investigational compounds, about 450 drugs in oncology. And we ask, is there anyone that can completely invert the pattern of regulation that maintains tumor state? Okay, either alone or in combination. And so far, we've seen that basically in every tumor we looked at, there's somewhere between 2 to 6 of these compounds that we already have in the clinic that can actually do that. And when we use them, for instance, in a transplant of the patient, or in some cases, even in the patient themselves, we've seen dramatic results with the full abrogation of tumor viability in vivo. Yeah, and that's exactly a good point. I mean, what we've been trying to do is to use one of those old-fashioned keys that were essentially flat. They had a single tooth and it was a flat tooth. You turn the lock and it opens. So it turns out that cancer is a much more complex lock and requires a lot of different teeth, not just one. And so the ability that we have to actually kill cancer cell by targeting only one protein, even if it's the right protein, it's limited. And it's really only worked in tumors that are almost completely 100% driven by the aberrant activity of that protein, like CML, for instance. I mean, most tumors targeting the oncogene only gives you a finite reprieve from the disease, and then eventually it will come back, it will relapse, and it will no longer be sensitive to the inhibitor. We're hoping that by targeting these more complex locks, where you have maybe the ability to target anywhere from 3 to 4 to 5 to maybe 20 proteins at the same time, you actually now put the tumor in an irreversible state switch from which they cannot no longer recover. And you're targeting mechanisms that are downstream from the mutation, so you're not offering the tumor the opportunity of escaping the therapy by basically selecting cells that have alternative mutations or alternative bypass mutations. So, of course, this is all theoretical. Well, it's very practical since we've seen it in functioning both in transplants from human tumors into immunocompromised mouse and also directly in patients, but we need to still run very rigorous clinical trials to determine that this actually works in practice on the long run. Yeah, I think so. Right now, a couple of reflections that I have is that one of the things that came out from this research is that we have, I think, to spend a lot more time in understanding at a more precise molecular level the effect that drugs have in different cancers. We tend to, if you want, simplify intuitively the role of drugs by saying, oh, this is a MEK inhibitor or this is an HDAC1 inhibitor. And that, unfortunately, drugs tend to have extraordinarily tumor-specific patterns of activity. We call it the mechanism of action of the drug. And really understanding more precisely how drug participate, if you want, in modulating the dynamic of the cancer cell and also of normal cells has to become a really more fundamental area of investigation, because we find very frequently through these studies with complete experimental validation that drugs that are thought to be exactly identical, essentially, for instance, 2 HDAC inhibitors or an HDAC1 inhibitor or 2 MEK inhibitors, have profoundly different behavior in the tumor, because in fact when you look at their complete repertoire of targets and effectors, it actually maybe overlap on what you consider to be the key target, maybe MEK or HDAC, but actually substantially different on the rest of effectors and targets. And this is really important because we may be able to prioritize among a number of potential MEK inhibitors the one that really works in a particular patient with a KRAS mutation, while the other MEK inhibitor may not work at all. And this is because of these field effect that these inhibitors have. They're not clean. They are very dirty small molecule. They hit a lot of different things in the cell. And by the way, they hit things depending on the context in which you put them. So I think the combination of that suggests that this new field, which we call systems pharmacology, really requires some very fundamental deep thinking. And this is an area, for instance, in which Darwin Health is very, very much a player, because we have developed assays to very precisely characterize compounds and are working with a number of companies to actually help them characterize their compounds pipelines.